Hello viewers, welcome to this particular lecture on microbiology. In this series of lectures, I shall be discussing different aspects of airborne fungi and the pathogenicity of different fungal spores. Now the air may contain different organisms. It may contain viruses, it may contain bacteria, it may contain fungi, both vegetative cell as well as spores. But actually the spores of fungi are disseminated more commonly, and they are disseminated along with the suspended particulate matter like the dust particle. So very often they come inside our respiratory tract or they adhere to the skin surface and they cause infection. So I shall be mainly restricting to different types of fungi and in what way they are pathogenic, being airborne in nature. In this particular lecture, I shall be mainly discussing the different strategies that fungal spore they adopt so that they can be ideally pathogenic, or at the same time I shall also mention the different characteristics of these fungal spores, and then I will try to discuss one by one major fungal species and their pathogenicity, right? So to start with, I shall be discussing the different strategies. You see, fungal spores may be cut off in chains, like you can see here it is cut off in chain in case of Conidia of Aspergillus, where the Conidia are cut off very easily to air current, or sometimes as in Phyllophora they are borne in chain and they can easily be cut off, so they can be straight away get into the air current and come in contact with human host or animal host. Or sometimes it may so happen that they are borne in multiple layers. So if the upper layer is just sloughed off, the lower layer is exposed and then it is sloughed off, and by the time the second layer is exposed, the third layer is already ready. So this multiple layer of spore can also be an ideal strategy of an airborne pathogen. And to be very specific, like this particular ascospores, you can see these ascospores are born within ascus, but at the same time they are protected with melanin, and this melanin is protecting them from UV and other natural calamity or natural hazard. So they can remain active, they can remain alive for a longer period in air. Now coming to some characteristic, as I have mentioned that these fungal spores, they do have a distinct definite layer of melanin, or they are melanized. At the same time they may have multiple layers. There may be an inner layer of muramic acid or chitin, which is a nitrogenous polysaccharide, followed by multiple layers of beta glucan, which is forming a mesh, and finally there is a polysaccharide mannose or polysaccharide of mannose, commonly called mannan. And at the same time there are specific receptor within this mannose residue which will make the fungal spore or fungal cell adhere to some specific surface, and definitely this multiple layer will make the fungal spore survive for a longer period in air. Another very interesting strategy is that they are having the multiple layer of glucan, and this may be, they may be having beta-1,3 glucanase or other enzymes which actually cut this glucan residue. The glucose residues are cut off, but the moment they are cut off by this glucanase enzyme, again they regrow. So that means what? If by any means, if by any other pathogen or any other hyperactive organism the fungal wall is destroyed, again they have the ability to resynthesize it. That means they have the ability to repolymerize their wall so that their sustainability increases in air. Okay? You can see that this particular fungal spore, very nice, they look like cricket ball, but their outer surface is having UV resistant layer. It may be due to melanin, but this UV resistant layer, you can see they are fluorescing UV. That means they are reflecting UV. So the UV ray cannot get inside. And you know UV naturally it is killing a pathogen or killing a pathogenic organism, but here the UV ray cannot percolate. So the fungal spore are not really killed by UV exposure. So that increases their pathogenicity. And another very important point which I was telling, that fungal spores may come inside, but they actually adhere to the mucous surface. You can see here the lung tissue or the tracheal tissue, they are having this fungal spore. They easily adhere to the mucous surface by the production of a polysaccharide adhesine. Now, commonly when we consider airborne fungal pathogenic spore, 3 common species are there, though I shall be telling you about other species as well. And these include the Alternaria. This Alternaria is having long chain of Conidia, which is multicellular Conidia, and typically they are sandal shaped, but they are produced in chain. And there will be typically the multiple sporulating Cladosporium, which are very commonly sporulating in nature, sporulating from multiple points. So even if one point is cut off, the other point will grow. And thirdly, this is the most pathogenic one, the acropetal generation of this particular Conidia from the vesicle of Aspergillus. And so dense is that growth that easily you cannot really get rid of this particular fungal spore. Now, to start with, a particular group of pathogenic fungi which is airborne, we will consider or stress on mucor. Now, mucor actually belong to a group of Zygomycetes or Zygomycota. So, it is Zygomycetes class, order is Mucoralis, family is Mucoracea, and the genus is mucor, and the species, most common species is Indicus. Though of course there may be other names given, that is Mucoroxy is another name. And this particular mucor fungi is characterized by aseptate mycelial mesh, that is a thread-like structure without septation or cross wall. And they are having specific airborne filamentous structure on which a typical ball-like structure is there. And this ball-like structure actually is known as the sporangium. The central part of the sporangium is actually not having any spore. It is called the columella. And the peripheral part is bearing the spore, which is called sporoplasm. So this is a reproductive structure. But at the same time, they sometimes may reproduce by cutting off from the tip of the hyphae, and typically this structure is called oidea. So oidea production is another way by which this fungal will reproduce. They do have chitin, but actually sometimes this chitin may get deacetylated, removal of the acetyl group, producing chitosan. Now, what way it is beneficial? It will help in sustainability. Chitosan content of the fungal mycelia will make it more stable. Okay. Now, coming to the different ways of reproduction, apart from this sporangium, apart from this oidea, they can reproduce sexually, and this actually is when two particular mycelia will come close to each other and they will produce two gametangia, and this gametangia will fuse, which is called gametangial copulation. And then they will produce the thick-walled spore, which is a diploid spore called zygospore. And the zygospore will allow the particular fungi to take rest for some time, and then they will germinate to produce germ sporangia, and this germ sporangia will produce the sporangiospores, and they will germinate to produce a new fungal body. So this is an ideal way by which they can sustain the unfavorable condition, and at the same time the spores will germinate and they will produce to greater areas with the advent of favorable condition. This is a very unique case where we can see that a particular mucor will show typical budding. So we can say that this is dimorphism in mucor. So mucor sometimes when it is kept in sugar solution, it will actually grow into yeast-like condition and it will produce bud. Profuse budding is there. So it is very often called yeast stage of mucor. Okay.
Sir, I have a question. If mucor indicus reproduces by gametangial copulation, then what is the utility of budding process?
Yeah, you see, I told you that budding will only be initiated when there is adequate sugar. So when they have sugar, that is the stimulus. So they will reproduce very fast so that they can utilize the sugar, and that's why they reproduce by budding. Okay, so that is why it is reproducing by budding. Now another very interesting point, it is showing the typical glucose metabolism, producing the phosphogluconate, producing xylulose 5-phosphate, and from there they can produce acetate, ethanol and lactate. So these are typical fermentation end product of glucose carried out by mucor. So that way mucor may be industrially important because it is producing organic acid or ethyl alcohol. But of course that is a separate story altogether. It is not coming to the way of pathogenicity. Another very interesting point is that if this is a typical mycelia of mucor, you will find that they grow very fast, so they require energy. and that also gives energy for pathogenicity. They have vacuole, and they also have oil droplets or oil content. So they are typically oil producing fungi. But this group of fungus, that is the Zygomycetes, which may include mucor, which may include Rhizopus, which may include the Cunninghamella, these are typically the same type of Zygomycetes fungi, and they are commonly called the grass, that is generally regarded as safe. So automatically they will not cause infection. They are opportunistic infectious organism. They will get in, they will remain, and then they will invade. And you can see that they grow normally in soil. So they can produce typical stolon-like structure as in Rhizopus. They will produce dense green spores as in case of the mucor or Rhizopus again. And sometimes these spores may be larger in size like in case of Cunninghamella. But in all the cases, these spores will get inside the living body, remaining there for some time, and sometimes they may behave as pathogenic organism. So let us see in what way they are pathogenic. They may get inside the mucoidal tissue, and you can see the typical biopsy of mucoidal tissue. This is the fungal mycelia. Or sometimes MRI of the brain may typically show these are the fungal mycelial zone. So fungal infection can be detected by MRI. Or sometimes typically there will be oral lesion. Apparently people will think that is a cancerous lesion. No, this is a fungal lesion, because if we test it, we will see that they are containing fungal spore. Sometimes they can invade the blood vessel, producing an inner layer, and thereby they can induce a situation similar to thrombosis. Or sometimes they may invade the sinuses of the head, paranasal and prenasal sinuses, and they cause similar symptoms as sinusitis. So apparently you will say, okay, I am suffering from sinusitis, but never really you will think that this is coming from an airborne fungi. Or sometimes, typically, you may suffer from boil, but this boil may be due to mucormycetes. And this is a dangerous picture. You can see that the fungal colonies has gone inside the lung and has grown inside the alveoli, creating specific lesion similar to tuberculosis. It's a dangerous disease. Now, how it can be detected? Nothing like isolation of the tissue, taking the fungal mycelia if you get one and seeing it under microscope. You can see this typical sporangia and you can definitely say, okay, this can be mucor. Or if you can grow it in a typical saborod agar medium, it will produce typical colonies like this. You can say, okay, this is a mold which can be mucor, which can be Rhizopus. So these are ways you can identify it. And finally, after identification comes treatment. So how can we treat mucor? Ideally, mucormycosis is a very, stubborn infection. It can be detected, but treatment takes long time. Ideally, it can be by using amphotericin B injection. Mostly injection is most effective. Or it can be by the use of posaconazole, azoles, which can be topical use as well. Okay? Sometimes, if it so happens that this particular fungal infection is reduced, but the slightest of fungal spore remain inside, like say for example, this is a fungal tissue which has grown inside the meninges in the brain. Then surgically, you have to go for surgery. You have to take it out, that this is the fungal mass which needs to be taken out by operation. And there lies the risk. If that fungal spore is not removed, it will cause dreadful fatal disease of meningitis. So that way mucormycetes apparently, which is not very harmful when it is in the skin, but when it is going inside the brain, it can be 100% fatal. Okay?
Sir, I have a question.
Okay.
What should be the ideal method of treating mucormycosis?
Mucormycosis treatment actually, I told you, can be with one drug. But if the fungus is showing resistance to a particular drug, then that drug has to be replaced. So you can use azole, you can use amphotericin, or sometimes you can use steroid along with this particular drug. So depending upon your necessity, you have to select a drug, and very often the antifungal drug may be accompanied by some steroid molecule. And in severe cases, it can be surgical even. Now coming to another very important group, the next higher group is Ascomycetes. Ascomycetes fungi, very common is the Unicellular Candida. This particular Ascomycota, actually is same as yeast. So they belong to Saccharomycetes. The family is Saccharomycetaceae and the genus is Candida, and there may be different species. The most common one is the albicans, but there may be other species as well, like the Candida parapsilosis. It can be Candida tropicalis or it can be Candida dubliensis. But all these species will have typical yeast-like character. What is that? Single cell with a large vacuole, which are distinct diploid nucleus and the ability to reproduce by budding. And of course, when I consider their distribution can be wide. It can be in soil, it can be in air, and whenever it is in the air, it can come onto our food material. And sometimes it so happened that this particular wine, this is a red wine, which is having this fungal mass. So just think that particular fungus, Candida, has grown inside the wine, so they can tolerate, withstand that pH. Or it can get inside our gut and they will reproduce there. So it will create a film on the endothelium of our intestine. So that way it can be saprophytic, it can be parasitic, it can be opportunistic, pathogenic organism. How to identify the characteristics? Typically, they will produce discrete white type of colonies. And when they are studied, they are typically yeast-like. Unicellular, oval or round colonies, sometimes producing chain. And this chain can be, ideally, it is not a mycelia, but it is mycelia-like. So we call it pseudomycelium. But when I am growing it in colonies, actually they may grow as round colonies, which are usually white in nature, or sometimes they may produce rod-like opaque colonies. Now specifically, this type of structure or this type of structural modification is due to certain phenotypic switching. Now this phenotypic switching can be because of the activity of this particular EFG1P protein factor. So if the WOR1 gene is active, automatically they will get into the opaque state. But if by any means this opaque state is getting into EFG1 activation, by any means, then they will get into the white mass. So in one way or the other, if I can activate the EFG1 protein, then the particular fungus become modified. So we call it a dimorphism, but this dimorphism will bring about phenotypic change, and this phenotypic change actually is changing the pattern of the fungus. Now one important question arises. Apart from this particular switching on and switching off of gene, there may be other ways by this fungus can modify. What are these ways? They can sometimes show chromosomal aberration, which can be deletion, which can be translocation, or which can be insertion. So different transposons are also there in particular Candida, which also increases their pathogenicity. And at the same time, sometimes they may show variable property of genetic code. Another very interesting point which comes to my mind is that they have chromosomes and they have mating factors. Both in yeast and in Candida, there will be specific mating factor, which can be factor A, factor alpha. So whenever there are two factors, there will be what is there? They come close to each other and they will produce heterozygosity. So in case of Candida, which is a diploid, it's a very important point. It is a unicellular fungus, but it is diploid in nature. They will develop heterozygosity. Like yeast, Saccharomyces cerevisiae will develop heterozygosity but in haploid state. So this particular heterozygosity, there is some advantage. What is that? They increase their pathogenicity. They increase their variability. They increase resistance to drug. So in all way, they are adding to their pathogenic nature. Okay. Apart from that, there may be some important factors which make them ideally airborne pathogen. They may include the typical HWP or the hyphal wall protein. And this hyphal wall protein actually is helping in the addition or colonization in the human host or human tissue. And the one which is having more expression of HWP, there will be more pathogenicity in that particular Candida. Similarly, there may be expression of the typical SLR or SLR gene, and this particular SLR gene actually will increase or decrease colonization within specific areas of the body. Though here I have considered kidney. You can see here, this is the wild type strain, this is the SLR recessive, and this is the SLR dominant. You can see the SLR dominant will show more colonization here in case of renal pelvis, but in case of renal tubule, they are localized, but density of infection increases. So what does it mean? That depending upon situation, the fungus also know that which particular protein factor I need to express so that I can be target specific pathogenic organism. Now we come to certain symptoms. As I told you, it is airborne, so it will come inside our tract. So very commonly it will invade our buccal cavity. So this is how it colonizes. You can see these white colonies are that of Candida in the tongue. It can be adhered to the skin and it can produce typically scaly appearance of the skin, or sometimes it may cause vaginal infection. This is the vulvovaginal infection. It can cause infection of the oral cavity inside the endothelium of the buccal cavity. It can cause, typically this is a deep vein thrombosis or thrombophlebitis, where there will be clot in the vein and thereby there will be typical reddening of the limbs, which is not a superficial infection. Mind you, it is inside the blood vessel. But of course the entry is through skin, or sometimes it may get into our GI tract causing infection. These are the colonies inside our esophagus or food tract. Just see the colony of Candida invading the gastrointestinal tract. Dreadful condition. Okay?
Sir, which organ is more affected by Candidiasis?
You see, apparently any organ can be invaded, but because the contact of epithelium or skin or mucus is more, so these external organs are more affected, and gradually they get inside the blood vessel and gradually it becomes systemic. But depending upon the situation, the more it spreads, more devastating it becomes. Now very commonly this particular Candida can spread with a very bad practice, that is piercing of tongue. Piercing of tongue can lead to Candida infection through contamination. So this is how you can see this tongue has been pierced and these are the colonies developing below the tongue. Because of this tongue piercing habit, there may be Candidiasis developing. And nothing like that, you know, piercing of needle is very much associated with HIV. So the moment HIV develops, HIV patients are more prone to Candida infection. Now how to treat them? Actually, because it is skin, it can be ideally by ointment, topical use of miconazole, which is ideal. At the same time, there may be some systemic drug, but I told you the more devastating it is not being treated by any common drug. So we can give some symptomatic relief where we can use clotrimazole with corticosterone. That is a steroidal drug. So combinational therapy is commonly carried out. And if it is actually causing deeper infection, there is no proper answer. So in this particular lecture, what we have studied the characteristics of different fungal infection. We have also studied the different ways by which they are causing infection, and also we have characterized two different fungi or group of fungi which include the mucor or which also include the Candida, the different ways by which they cause infection.
Sir, does antacids and antibiotics have any effect on Candidiasis?
Yeah, it's a very good question. You see, antacids, they will reduce the HCl level. The moment it is reduced, fungal spore will not be killed by HCl. So there will be more invasion in that gut. At the same time, when you are taking antibiotic, there are some friendly bacteria in your gut which will be killed. So the moment they are killed, their inhibitory action on Candida will be reduced. So there will be more Candidiasis. So apparently these drugs can actually be more helping or helpful in the development of Candidiasis. But of course, in immunocompromised patients or in HIV infected patients, this Candidiasis is becoming dreadful and sometimes fatal. So we should be cautious about airborne fungal infection. Thank you.